Hello everyone and welcome to my channel Be Yourself. This is Dr. Nisharma and today I'm going to deal with the answer keys as well as explanations for the CSIR-JRF-NET Life Sciences of Part C, which was held on June 2015. So let's start. I already have discussed the Part B question. This time I will discuss for the Part C. Let's start with the first question. The question is, the sting of a bee causes pain, redness and swelling. Melittin is a major peptide in bee venom. Melittin is the membrane binding peptide that is involved in activating phospholipases in the membrane. The possible target phospholipase that is activated by melittin is actually melittin activates target the phospholipase A, which generates the arachidonic acid. So the answer will be 2. Not only bee venom, rather snake venom also target phospholipase A2. Apart from the generation of arachidonic acid, melittin also releases amylase, changes the membrane fluidity, and also releases linolenic acid and also help in the synthesis of prostaglandins. So these are all the function of melittin induced functions, and I have discussed this because I already have discussed while I was discussing on the December 2016 question papers that if you will analyze the past year questions, then you will come to know that the pattern of question is almost similar. So you will analyze that similar topics are asked all the times, just they just twist the questions in a different manner. So better prepare the related questions from the past year question. Move to the next question. The glycolysis and citric acid cycles are important pathway to generate energy in the cell. Given below are statements regarding the production of ATP. The question is quite simple. If you have a general idea of the glycolysis and citric acid cycle, you will come to know that during this cycle, there are in total 10 molecules of NADH and 2 molecules of APDH2 is produced per molecule of glucose. Another thing is that the coenzyme produced are oxidized by the electron transport chain. So here the answer will be A and C. As per this, the answer will be number 4. Move to the next question. In a 30 residue peptide, the dihedral angles phi and psi have been determined by one or more methods. When their values are examined in the Ramachandra plot, it is the first point is not possible for the phi and psi values to be distributed in the helical as well as beta sheet region. It is possible because the phi and psi angles is it does about the difference between psi and C bonding and C and N bonding. So actually what happened, it generally had give the idea of the steric hindrance between the C and C group and C and N group. So it can for each amino acid there will be variable in the psi and phi, and it treat for the each particular amino acid. So it is possible that the values can be distributed in the helical as well in the beta sheet region. So the statement is wrong. Next point is possible that psi and phi values are all in the helical region. It may be possible, but here another point is that all the circular dichroism spectral studies indicate beta sheet conformation. It is not possible because it's also tell about the Ramachandra plot, also tell about the structure of the protein with the help of psi and phi angles. So if it is telling that it is in the helical region, that it is not possible that the protein will be the beta conformation state. So this statement is also wrong. Move to the next point. Possible to conclude that the peptide is composed entirely D amino acids. It's possible because there are different reasons and the quadrant of Ramachandran plot which tells about the D and L structure of the amino acids. So the statement is right. Now, another point is that in about the Ramachandra plot, I already have discussed in the explanation of December 2016. You can also refer that video too for more information about the Ramachandra plot. I'm not repeating same thing again and again because it will waste my time. So, move to the next question. Hydrogen bonds in proteins occurs when two electronegative atoms compete for the same hydrogen atom. This is the donor and this is the acceptor one, and this is the hydrogen bonding. The angle theta between the donor and acceptor of a hydrogen bond was determined from the large number of axis structure of protein as shown below. Actually, the theta angle between the donor and acceptor generally varies between 90 to 180 degree, or if more appropriate at the 160 degree. So if you will look the all the figure, if you will look A, B and C graph, then come to the graph number C. Here it's 50. It's come. It will be 60, 70, 80, 90, and finally it's 20. You will come to know that this is more appropriate as per the data, which is the 90 to 180 degree. But if you will see this angle, then it's far beyond the 90 degree, and this also it's far less than the 160 degree, which is the most appropriate angle between the donor and acceptor. So the answer will be only C. As per this, the answer will be number 3. Next question. In the accompanying figure, reaction kinetics of three proteins A, B, C is represented. Protein concentration used to obtain this data are: A has a concentration of 1 mg per ml, B has a concentration of 4 mg per ml, and C has 2 mg per ml. Next point is here. If catalytic efficiency is defined as Kcat by Km, Kcat is the turnover number and Kcam is the Michaelis-Menten constant. Which of the following statement is correct? Now, come back to the graph. Here it's showing the Vmax for the B, Vmax for A, Vmax for C. Let's extend it to get the value. I am taking it as approximately as 18 as a Vmax, and for C, I am taking approximately as 12. I am taking this in the value because I can't take exactly what it is because there is no bar here. So I am taking a estimated value here. I have taken 18. You can also take 17, 19 for A. It up to you, but I feel that taking 18 as a value will be easy for the calculation for me. So I'm taking here it as 18. So here we have to calculate Kcat, and I have already told you that Kcat equals to Vmax by E total. The E total is given for different A, B, C concentration is given here. Vmax I have predicted here. I'm taking it as 30 for the B. So we have value for B, A and C. Now what is Km? We can also relate Km as equals to substrate when V is half of Vmax. Okay, so we can say that Km is equals to substrate when the velocity is half of the Vmax. So if you will analyze this, then you will come to know that in all the cases approximately at 1 micromolar, the Vmax is half of it shows the value of Vmax, and so we can say that Km for all the values, all the proteins A, B and C will be equals to 1 micromolar. So in this case, the main determining factor for this efficiency is basically Kcat. If the Km for different proteins A, B and C will be different, then we will consider the formula Kcat by Km, where Km will also affect the efficiency. But here, as the Km is constant for all the ABC, so we can neglect it, or even if you will divide with the same factor 1, then we will get the same result. So I am only calculating the value for K cat. For A, it will be Vmax, which is 18 by E total. E total is, for A, it's 1 milligram per ml and it's equals to 18. For B, it is 30 and the E total here is 4. If you will calculate it, it will be somewhat 7.5. Now for C, here Vmax is 12 and E total is 2. So it will be 6. Now arrange it accordingly as per the efficiency. Even if you will divide it with 1, then you will get the same value. So I am here ignoring Km. Then what will you get? Here 18 is greater than 7.5 is greater than 6. So the answer will be A greater than B greater than C. It means the efficiency of A is highest, followed by B and C. Hope you understood the question. Move to the next one. Lipid rafts are involved in signal transduction in cells. Rafts have compositions different from rest of the membrane. Rafts were isolated and found to have cholesterol to a sphingolipid ratio of 2:1. The estimated size of the raft is 35 nanometer square if the surface area of cholesterol is 40 Armstrong square and a sphingolipid is 60 Armstrong square. How many cholesterol and sphingolipids are present in one raft? Here the ratio of 2:1 is given. If it means if sphingolipid is 1, then the amount of cholesterol will be 2. So now we can say that if the amount of sphingolipid is X, then the cholesterol amount will be 2X. Now take another page to solve it. Here we have given that size of raft is 35 nanometer square and cholesterol is 2, sphingolipid ratio is 2:1 and I have taken it as 2x and x, and the area of cholesterol is 40 Armstrong square and that of sphingolipid is 60 Armstrong square. Now the total number of cholesterol and total number of sphingolipid, when we will sum this, then we will get the area of the raft. So sum it out and equivalent and get it equal with that of the raft area. But one thing is there, here the unit is in Armstrong square and here it is in nanometer square. So better convert it into Armstrong square, which will give 3500 Armstrong square. As here it is square, so I have multiplied with 100. So 2x into 40 Armstrong square plus x into 60 Armstrong square will be equals to 3500 Armstrong square. Now when you will solve this, you will get the value of x as 3500 divided by 140. You will get it as 25. So here the value of x is 25, which means the number of sphingolipid will be x equals 25 and that of the cholesterol will be 2x equals to 50. So the answer will be, come back to the question, the answer will be 50 cholesterol and 25 sphingolipid. So simple it was. Now move to the next question. Glycolipids and sphingomyelin are produced by addition of sugar or phosphorylcholine to ceramide on cytosolic and luminal surface respectively of the Golgi apparatus. It is added during the post-translational modification. Finally, after such modifications, these molecules are located on the outer half of the plasma membrane. What key events are responsible for such localizations? While I was discussing about the endoplasmic reticulum and the related question with that, I have discussed that the one which is expressed on the, suppose this is Golgi body, it's not well drawn. I am drawing it here. It's the Golgi body and this is the cell. This is plasma membrane. Suppose it's the plasma membrane. It's the cytosolic side and it is the luminal side. When any modification will be on the cytosolic side, then what will happen? It will bud out from here, and in the form of vesicle, it will fuse with the plasma membrane. But when it will be on the cytosolic side, it will always remain on the cytosolic side. So you can also draw it like this. This will bud out in the form of vesicles, and when it will fuse with the plasma membrane, it will be exposed on the cytosolic side. But when it will be on the luminal side, it will be when it will form a vesicle, and when it will fuse with the plasma membrane, then the content of the vesicle will be excreted out, and so it will be expressed on the outside of the plasma membrane. But here another point is written here is that what is gone, it's expressed on the outer half of the plasma membrane. It means if this is a double layer of the plasma membrane, then it will be expressed on the outer membrane. It means it has flipped from the inner membrane to the outer membrane, because we know that the modification or the protein get inserted on the on the cytosolic side of the membrane, and then it undergo flippage. With the help of flippage activity, it flip from the cytosolic side toward the luminal side. So there will be two actions. One will be the action of the flippage, and other will be the membrane fusion, because of which the protein will be expressed on the outer membrane of the plasma membrane. So the answer will be number 2. Question number 78. In an attempt to study the transport of secretory vesicles containing insulin along microtubules is cultured pancreatic cells, how would treatment with colismid affect the transport of these vesicles? Actually, colismid has two functions. One is that it depolymerizes the macrotubules, and other is that it will affect the ER organization. So as per this, the answer will be number 2, because here the polymerization will be inhibited by the colismide, and another thing is that if it is affecting the endoplasmic reticulum, which is involved in the secretory pathway, it will also inhibit the transport of secretory vesicles. So the answer will be number 2. Next question is that when cellular plasmid having centromere sequence are transformed into yeast cell, they replicate and segregate in such each cell division. However, if a linear chromosome is generated by cutting the plasmid at a single site with the restriction endonuclease, the plasmids are quickly lost from the yeast. It is known that genes on the plasmid are lost because of the inability of the chromosome ends. What would be done so as to restore its stability and can be inherited? If here end is affected, to restore end, what we do, we can incorporate the telomerase sequence to the end of the plasmid, so that the sequence which is lost during the cutting will always be preserved. So the answer will be number 3. Next question, question number 80. REC8 is a meiosis specific cohesin that maintains centromeric cohesion between sister chromatids in meiosis 1. Which of the phenotypes listed below would you predict will be manifested in a REC8 deficient yeast? Actually, if the cohesin protein is there, it holds the sister chromatids at the centromere position. So it will be somewhat like this. The cohesion protein will be on the centromeric side, and it will be on the 4 sister chromatids will be here. So as meiosis 1 is responsible for the reduction division, because of the inefficiency of the centromeric cohesion, there will be improper reduction division, and this will decrease the viability of tetrad. So the answer will be number 2. Next question. Eukaryotic DNA polymerase alpha has the tightly associated primase activity but moderate processivity. If there will be moderate processivity, then there will be very short polymerization of DNA, but it is not so in case of eukaryotes. So here polymerase alpha cannot go for the polymerization of the whole length of the DNA in case of eukaryotes. Next point is that DNA polymerase epsilon and delta are highly processive but lack primase activity. If it don't have primase activity, then it is also not possible for the replication to proceed, because DNA polymerase can't proceed without without the addition of primase. So it can't alone can help in the replication process in case of eukaryotes. So there must be primer synthesis. So after the activity of alpha, it is only possible that either epsilon or delta will act. The next point is here, given below are the four statements about leading and lagging strand synthesis in eukaryotes. Which one is true? Before answering it, I will discuss this point so that you yourself be able to understand the question and be able to answer it, and you feel definitely confidence that you have solved this problem, because some questions are so easy, but because of lack of certain tiny things in your mind, you miss the question or go for the negative marking. Here it is mentioned that alpha is involved in the primer synthesis both for leading and lagging strand as per the question. Next is that beta repaired the DNA similar, and it is similar to that of the polymerase 1. Gamma especially for the replication of mitochondrial DNA. And here DNA polymerase delta is responsible for the replication of leading strand as well as involved in the proofreading activity. Now move back to the question. Here there may be possibility that once the primer is synthesized with the help of polymerase alpha, then there will be followed by the polymerization switching of either epsilon or delta. So the here answer will be number 4. Next question is, in context of lac operon, if two bacterial strain P1 and P2 with the genotypes OCI plus Z minus and O plus I minus, this is the inducer, this is operator, and Z code for the beta glucuronidase activity respectively, were used to produce marrow-diploid daughter strain D, which one of the following statement correctly predicts the expression of Z gene? Z gene is only possible when the Z is in the positive form. So there will be no expression of beta glucuronidase activity in case of P1, and for the P2 it will always be in the constitutive expression because here operator is on. So P2 will have the constitutive expression, and the D, which has the possibility that either of the sequence or this sequence or the recombinant of this can be present. So D can express the beta glucuronidase activity when there will be the induction of inducer or operator to function Z as a positive. So it will be inducible expression. So as per this, if you look the option will be 1, where P1 has the no expression, P2 is the constitutive expression, and D has the inducible expression. Next question, in order to study the transcription factor 2H, it was cloned from a large number of human subjects. Surprisingly, the subjects having the mutation in transcription factor 2H also showed defects in their DNA repair system. Actually, transcription factor 2H is involved in the nucleotide repair system. Because of this, if we will mutate the transcription factor 2H, there will be also defect in the DNA repair system. So as per this, answer will be in the mammalian system, replication factor 2H plays an active role in transcription coupled DNA replication process. So the answer will be C. I have discussed about the function of transcription factor 2H in my video in my channel. Be yourself under the topic RNA transcription. You can watch from that. Next question is, the 3' end of most eukaryotic messenger RNA is defined by the addition of a poly-A tail, a processing reaction called polyadenylation. We all know about the polyadenylation. Actually, what happened when DNA replicate, it replicate even beyond the site of the polyadenylation. This is the site where the polyadenylation is to take place. Then what happened? Then the polyadenylation site is marked by AAA U AA on the 5' side and GG and GU and UU and G sequence on the 3' side. After that, with the help of endonuclease activity, hydroxyl is exposed, and with the help of enzyme polyadenylase polymerase, ATP is converted. It will have to convert in the AMP form to get the 3' end polyadenylated. And now we have the matured RNA. So as per this, the question answer will be, move back to this question. It will be the poly-A polymerase is a template independent enzyme. Yes, it is a template independent enzyme because we don't prefer any template as a reference where poly-A is to take place, because here there was a marker of triple A and toward the 5' side and 3' side. So it is recognized with the help of a marker sequence, not with the help of any template. So the statement is true. Next statement is addition of AMP from deoxy-AMP. It was not there, only AMP was there. So the statement is wrong. Poly-A polymerase is RNA template dependent. No, it's not RNA template dependent. Next point is poly-A polymerase catalyzes the addition of ADP, not ADP, rather it catalyzes the formation of AMP from ATP. So the statement E is also right, where it says that the poly-A polymerase catalyzes the addition of AMP from ATP to the 3' end of the messenger RNA. So the answer will be number 3. Next question is, with an intention to identify the gene expressed in an organism at its specific stage of development, messenger RNA were isolated from the given organism, cDNAs were synthesized, cloned in a suitable vector and sequenced. A few of the cDNA sequence showed no matches with the genomic DNA sequence. Further, it was observed that these sequences were U-rich and found to be in stratus dispersed along the sequence. The following may be the possible reason for appearance of such RNA. Actually, this particularly take place in trypanosoma by the process of trans-splicing, where guide RNA, also known as slRNA, introduces U's involving endonuclease and terminal U transfers and RNA ligase. Then it will lead to the deamination and it will convert C to U. Look how it happens. Actually, for this answer will be C, D and E. Actually, it is particularly in trypanosomes where there will be a nucleophilic attack of hydroxyl of adenine toward the J, and after that it will form a Y-shaped complex. After that, the hydroxyl will also attack on the exon part and it will be spliced out, and where then it will form the these types of splicing is known as trans-splicing, and it particularly takes place in trypanosomes. Next question is, when one isolates ribosomes from the bacterial lysates, apart from 70S, 50S and 30S ribosomal subunits, one also finds a small population of 100S, 130S and 150S subunits. EDTA dissociates these larger ribosomal subunits into 50S and 30S, suggesting that they have both the subunits. Upon addition of cations, they reassociate into 70S, but none of the other form would be detected. What is the reason for not obtaining more than 70S forms? Actually, these are the form of the polysomes which are formed by the integration of the individual 70S, and it is not possible in the de novo situation to reassemble these polysomes into the 130S or 150S. So the here answer will be the 100S and 130S extra represent polysome that cannot be reassembled de novo without other cellular components. This question is little bit tricky, but it's quite easy. If you understand the question very easily, you can easily solve such types of questions. It's quite simple because answer is there itself in the question. So let's see the question. Certain chemokines are known to suppress HIV infection, whereas pro-inflammatory cytokines are known to enhance infection. In order to explain these findings, control and chemokine receptor knockout animals were treated with a pro-inflammatory cytokines followed by HIV administration, and then infection was assessed periodically. Which one of the following graphical representations given below best explains the experimental results? Here, in control, there is a receptor, and in the knockout animals, receptor is removed. So, when we will treat with HIV virus, then what will happen? The one which don't have the receptor, it will not able to infect inside the cell. Rather, it will remain in the blood. So the one which is the which have the knockout receptor, the level of viral in the will be remain in the blood, but in control, which is controlled by pro-inflammatory cytokines, and it will enhance the infection of virus, and it will be engulfed by the internalized in the cell. It will not remain in the blood. So the amount of viral in the blood will be lesser in control in compared to that of the chemokine receptor knockout animals. So the answer will be number 1. Quite simple question is here. Of the following signal processes, which one is not involved in cellular movement or cytoskeleton changes? It's definitely number 4. A researcher was studying the protein X, which has been observed to move across the cell when an extracellular electrical stimulus is provided. An artificial peptide P was prepared which resembles the structure of connexins and competitively inhibits connexin formation. Which one of the following statements will best explain the fate of protein X if the cells are treated with peptide P and then electrical stimulus is provided? Actually, connexins are responsible to maintain the proper functioning of the gap junctions. If it is not be prepared properly, then it will definitely fails to move across the cell due to improper formation of gap junctions. So the answer will be number 2. Next question is, the patient with breast cancer was given a dose of radiation along with chemotherapy and was apparently cured of the tumor. After 5 years, a tumor was noticed in the patient's lung, but the doctor confirmed that it was derived from the cells of mammary gland. Actually, what happened during the chemo treatment, there will be some residual remains inside our body, and this can also infect to the nearby organ. So this might be possible that there some resistant cells or the residual cells may have migrated from the mammary glands and result in the tumor in the lungs. So the answer will be B, and sorry, here it's another option is here, so it will be only B. Move to the next one. Chromatin condensation is driven by protein complexes called condensin, which are the membrane of the family of structural maintenance of chromatin. Proteins that play a key role in the organization of eukaryotic chromosomes. Condensins, along with another family of SMC proteins called the cohesin, significantly contribute to the chromosomal segregations during mitosis. If cells are treated with the inhibitor of CDK1 phosphorylation immediately before the cell enters the M phase, which of the following statement is most likely to be true? Actually, what happened during the progress of to M cycle to metaphase, cyclin CDK1 and cyclin B actually leads to the chromatin condensation by phosphorylation of condensins. Another point is that it also involved in the nuclear envelope breakdown and fragmentation of Golgi apparatus as well as spindle formation. So when just before entering into the M phase, this CDK1 cyclin B activity is stopped, then what will happen? It will prevent the condensation of the chromatin. And what happened for the condensation of chromatin? We have condensin protein is there, which condense the chromatin into chromosomes. But if these condensins will not be there, then what will happen? It will not, first point is that it will not able to condense the chromosome, and second point about the condensin is that it is synthesized just after the dissolution of cohesin proteins. So the role of cohesion is that it helps to bind or the proper alignment of the chromosomes, and it will bind all along the length of the chromosomes, and it get dissolved all along the length. Only it remain in the centromere regions where it hold the chromosomes for the equal distribution of chromosomes. But when once the CDK1 activity is stopped, then this cohesion will also not get resolved and the entire length of the chromosome will remain together. So the answer will be that the sister chromatin are held together by condensin, or it will be held together by condensins along the entire length of the chromosome. So the answer will be number 2. This question is also quite simple. The question is that there are three substances A, B and C. Given below are the pattern of immunological responses in rabbit where A is administrated along with C, B is administered along with C and E is conjugated with B and then administered along with C. Here now if we will look the response for the anti-A, it's quite parallel to that of the time. So it means it doesn't induce antigen against itself if, when it is administered along with the C. So it can be a protein or the large size protein which haven't undergone processing so that it can activate antibody against itself. Graph number 2 has the anti-B activity. So it may be happens which has the ability to induce the activity of antibody against itself. And in number 3, here it is that A is conjugated with B. It means the protein is conjugated with the hapten which has induced the antibody formation activity and administered along with C which may be adjuvant which has relatively increased the activity of its antibody formation. So as per this, C is adjuvant, B is the haptens and A is the protein. So the answer will be number 1. Next question is, following are certain statements regarding seed development in plants. During final phase of development, embryos of orthodox seed becomes tolerant to desiccation, dehydration, losing up of 90% of water. Actually, orthodox is seeds which is very tolerant to desiccation, and even if it is 5% hydrated, it can easily come over the dormant stage, or rather it doesn't pass the quiescent or dormant stage during its development. And such types of seed is also known as precocious germination, and this is a statement also seems to be correct, as the statement is precocious germination is the germination of seed without passing through the normal quiescent or the dormant stage of development. So here orthodox seeds which can even germinate in the 5% of hydrated conditions. Such types of seed is also known as precocious germination, but this activity of precocious germination is inhibited with the help of abscisic acid. So the statement A, C and D is right. So the answer will be number 4. Next question is, development of vulva in C. elegans is inhibited by the induction of a small number of cells by short range signal from the signal inducing cells. Which reference of this following statement is put forward? Actually, vulva formation in C. elegans is controlled by the anchor cells. If the anchor cells activity will be stopped, then there will be no formation of the vulva. So the statement here, when the anchor cells are abated early in the development, no vulva will be formed, is true. Next point is that even there will be the signals or the inhibitory signals from the hypodermis region, then it also will affect it will inhibit the development of primary or secondary vulva formation. The signals here for the anchor will be given to the anchor cell, and this is known as LIN39, and it also induces the secondary fat in this cell. So the answer A and C is true. So the answer will be number 2. Next question is that, which one of the following about the development of sea urchin embryos is true? The answer will be number 1 is from the developmental biology, and here it is that during the development of sea urchin, each blastomeres of the 4 cell stages processes a portion of a original animal vegetal axis, and if isolated and allowed to develop will form the complete but smaller size larva. Next question is, what will happen if wingless RNAi is expressed in the wingless expression cell from the stage when this gene initiates its expression in the developing Drosophila development? Actually, wingless is stimulated in the posterior region of each segment or in the anterior portion of each para segment. But if RNAi is present there, then it will not allow the wingless to be expressed. So, as per this, the answer will be the enhanced progression of wingless scouts will broaden, it will not broaden the area, rather it will inhibit. So, as per this, the posterior compartment of each future segment will get affected. So, if RNAi will be there, then it will affect the posterior compartment of each future segment will be affected, as the wingless is expressed from the posterior region of each segmented section. Next question is, here are certain terms. First, let me explain about the invagination. Actually, invagination is particularly, if you will see, it's in the intestine region, there is the invagination of villi-like projection. It is particularly from the endodermis region, and involution is the, if we can say like this, if this is the outer layer of the cell, then the second layer will be grows just below this layer. There will be inward projection of the cell. The ingression says that there will be, let me draw the ingression here. If this is the layer of the cell, then the second layer of the cell will be involved in this manner. It will be lifted upward. And the delamination is the here the delamination itself, the name indicates that there will be lamination or the formation of different layers during the embryo development. And epiboly has expansion of one sheet over the another. So as per this, if you will look the number one is invagination. Invaginations is the not splitting, it is the infolding of the epithelial layer. So invagination go with the number 3. Here if you see 1 is not, B is not 3, it should be 3. So the answer option 1 is wrong. Let's see the involution is the 2. Involution means the splitting of one cell is not a splitting, it's the movement of epithelial cell as a unit of close deeper the layer on the embryo. Answer is number 3, because if you will see, it's only have the correct matches in which there is ingression, and ingression is the migration of individual cells from the surface into the anterior of the body. As I have told that this is the outer layer of the cells, and there was the migration of individual cells toward the anterior of the body. So sorry, of the embryo, and it is shown by the mesodermal in sea urchin. So the answer will be number 3. Formation of digits and sculpting the tetrapod limbs requires death of specific cells in the limb in the programmed manner, which is also known as apoptosis. Which one of the following interactions could explain proper limb formations? This is quite simple. That is actually this is the pathway where the apoptosis is induced with the help of WNT and beta-catenin. Next question is, light is the important factor for plant growth and development. There are several photoreceptors in higher plants such as Arabidopsis thaliana involved in perception of various wavelengths of light. Some statements are given below related to the photoreceptor. First statement is, red light photoreceptors are represented by a gene family. It's true. Phytochrome C is most prominent photoreceptor to perceive red light. No, it's the phytochrome B which is involved in the perception of red light, and A is involved in the perception of red light. The statement is wrong. Cytochrome 1 and cytochrome 2 have evolved from the bacterial DNA photolysis. The answer is true. And far red light is perceived by phytochrome D. I have discussed that it is perceived by D. So the statement is wrong. So as per this, answer A and C is true. So the answer will be number 4. Last question of this video is that pyruvate dehydrogenase is the subject to feedback inhibition of its production in glycolysis. Some of the chemical compounds which might be involved in the process are listed below. Actually, the one which is the end product of pyruvate dehydrogenase will act as a feedback mechanism inhibitor for the pyruvate dehydrogenase. So we know that the NADH and acetyl coenzymes is the end product from the pyruvate to when it will get converted with the pyruvate. Let me write here pyruvate to acetyl coenzyme A, then NADH and acetyl coenzyme is produced. So if this is present there, then it will inhibit the activity of pyruvate dehydrogenase. So the answer is A and C. As per this, it is 4. So I think it's enough for this time. In the next video, I will discuss the rest of the question of the part C. So don't forget to subscribe my channel, be yourself, and definitely share it so that more of you can benefit it, and click on the bell below the video so that you can get the notification as soon as I will upload my videos.